In human history, there are no old and reckless mushroom foragers, because those who were reckless never lived long enough to grow old. Ever since humans left the grasslands and entered the forests, a silent war has unfolded for billions of years, not between humans themselves, but between plants, fungi, and animals. One side evolves toxins for defense, the other sharpens senses and knowledge to survive. Every knife cutting into a mushroom stem, every cautious taste in a deep forest, is an evolutionary gamble between instinct and understanding. In that race, fungi are hybrids of life and death, neither plant nor animal, but ancient remnants from an era when life had not yet drawn clear boundaries. Along the edges of North American forests at night, a faint green glow sometimes rises from rotting wood. It is not magic. It is Omphalotus illudens, a bioluminescent mushroom. Its glow attracts insects, luring them into carrying spores farther away. Yet the same light has misled humans, who confuse it with chanterelles, edible mushrooms that look strikingly similar. Both are orange. Both grow near tree roots, yet differ in one detail: light. To ancient observers, glow suggested divinity, not danger. Biologically, the luminescence of the jack-o'-lantern mushroom comes from a luciferin-luciferase reaction, similar to fireflies. The light does not warn predators. It serves reproduction, a strategy to spread genes through attraction. It is proof that in nature, beauty does not equal safety. Light can be bait, and color can be an evolutionary lie. From Europe to Asia, humans have long carried traditional rules for identifying mushrooms. In
Our
Asia,
ancestors
people
knew
say,
the secrets
if insects
held
eat it,
within these fungi.
it's safe. In Europe,
Now,
if a silver spoon
we
doesn't turn
unlock
black,
them for the future
the mushroom
of
isn't
humanity.
poisonous. Yet both beliefs fail at the level of chemistry. Amatoxin in Amanita phalloides,
Just as I feared.
for example, does not react with
lord, I
silver because
swear
it
I
is
did
not a metal
nothing.
or sulfur compound.
As
It
you
is
can
a
see, the
stable
lemon juice does
peptide
very
chain,
little to the oxidation.
chemically indifferent
but with
to
the right
silver,
chelation agent, the
iron,
iron is restored instantly.
or flame. Insects often remain unaffected because their enzyme systems do not rely on RNA polymerase II, the very target amatoxin attacks in human cells. During the Middle Ages, many poisoning cases were recorded from Roman Emperor Claudius to royal courts in Russia and China, where white mushrooms sometimes became tools of political intrigue. Yet most victims were not assassinated. They simply made fatal mistakes, trusting folk tests that offered only the illusion of safety. These traditions survive not because they work, but because they give people a sense of control over nature, even when that control is imaginary.
The
Look
ribbons
at the
haven't
global
failed us yet.
distribution of
This is the
Galerina
path.
marginata.
It looks dangerous. Are you
And
sure?
something striking appears.
We lost the signal.
It exists on
We
every
stick
continent
together
except
and follow the
Antarctica.
marker.
A tiny mushroom less than 3 centimeters wide, yet carrying toxicity comparable to the death cap. Galerina produces amatoxin as well, but evolved it independently from Amanita. This reveals an important truth. Deadly toxins are not accidents. They are defense strategies selected repeatedly across evolutionary history. While plants developed bitter alkaloids to deter herbivores, fungi evolved peptides to deter gatherers. Ironically, this similarity causes hallucinogenic species such as psilocybe to be mistaken for deadly lookalikes. Some people searching for spiritual experiences instead begin a final journey from which they never return. At the molecular level, amatoxin does more than protect fungi. It shapes human behavior. It forces us to observe, remember, and teach, creating a form of cultural genome. Every death from a poisonous mushroom becomes another line of data etched into humanity's evolutionary memory. For thousands of years, humans relied on sight, smell, and taste to choose mushrooms. Yet those very senses have killed more people than many predators ever did. Only in the 19th century, with the birth of mycology, did humanity gain a reliable scientific tool, the spore print. Place a mushroom cap on white paper overnight, and millions of spores fall, forming distinctive patterns: brown, black, purple, white. No species shares the same print. It is a biological identity card. Impossible to fake through color or shape alone. Through such methods, science has identified more than 14,000 mushroom species and recognized over 100 highly toxic ones. Today, DNA barcoding algorithms can identify a mushroom from a fragment of genetic code only a few dozen characters long. Yet behind this technology lies a hidden cost: countless unnamed foragers throughout history. Prehistoric experimenters who tested nature with their own bodies. Knowledge of fungi is not merely a hobby for biologists. It is a survival legacy of humanity. Every identification technique, every classification handbook, is written upon biological failures, reminders of human limits before nature. The war between humans and fungi has never truly ended. Today we cultivate mushrooms in laboratories, extract antibiotics, and produce food. Yet deep in the shadows of forests, new species continue to evolve, quietly writing the next chapter of a biological arms race billions of years old. Because in the game of evolution, nature never signs a ceasefire.